old enough for her. She'd have to be at least 60. I'm sorry, it's true. All right. Here, there's not a lot to go over with this. Boys and girls, ladies and gentlemen. There are ladies here today, gentlemen. And where did I put this thing? I had the other, that MCAT thing. I'm going to do it this way. Yeah, I looked it over today. There's not enough. It was all about the eye and some odds and ends. It's not, what am I doing? Not fair. Sorry for this. Here we are. I meant to take the book with me, but I have a couple of questions at the end. It'll take 5 minutes to go over. And what's interesting about the eye, okay, a couple of things I gleaned from it, because you can read as well as I can. I think you do need to know the structures. Passing through the cornea, we know it's the clear portion of the outermost layer, even though it's just a realignment of the structures. The sclera and the cornea are the two most outer, the outer tunic. It's bent and refracted as it passes through the cornea, acts as a lens, okay? And then the cornea, as you can see with the sclera, the next layer, the so-called, I always called it the choroid, is that pigmented layer that absorbs the light, has a lot of the vascular supply. And then deep to that, the third, again, 3 layers, we consistently see that. The lining layer, in this case, is really the functional layer, which is the retina. And they explore that a little bit. That's there. The light's bent 3 different times, once from the cornea, once entering the lens, once exiting the lens. And then the whole idea, if your vision is correct, is going to focus here, what's called the macula, and particularly at that central area, like if you guys have corrective lenses, or like I do, at the fovea centralis, which is the optimal position. That normal vision would do that, and all of those. So you can see the diagrams here, the anatomies in the text. It's fairly easy. So, I mean, other than knowing the basic anatomical structures, the opening being the pupil, pigmented area, which is an extension of the choroid, the iris. Again, the sclera is merely structural and an attachment for the small muscles that allow us to move the eye. The optic nerve exits, and since all of the nerve endings come from it, or lead to it rather, where the confluence is called the blind spot, because there's no receptors there. And that in itself is, and you'll see that the nerve, in this case, the neurovascular bundle, the nerve and the blood vessels are on the interior, rather than like in a blood vessel where we see that stuff on the exterior, which is interesting anatomically, I think. Okay, and then they have the humors, the aqueous humor, minimal in the front, basically separating and creating sort of a distinctive environment here, as opposed to behind anterior, with the lens being the divider. And posterior to it, of course, is the vitreous humor. The vitreous we're born with. The aqueous is constantly made and drained. Obviously, if there's a problem with overmaking or insufficient drainage, that's glaucoma, something that we can address. Usually, sometimes it's even surgical, and there's a variety of medical ways to address it. The vitreous is formed at birth, really maintains the pressure that consistently keeps the retina appropriately in contact with the other layers, so it doesn't separate or detach, is the name frequently given to it. So the aqueous and the vitreous that are there, they're different. The vitreous is sampled a lot forensically, which might be an interesting side question, because it's there so that you can pick up little things, little information from there in a forensic or post-mortem standpoint, which is interesting, like that. So the vitreous and root, the chamber, again, vitreous humor, they're using the old term. And then finally, we get the actual retinal cells. And what's interesting about this is the light actually passes through those cells on the way to the rods, rather than the light immediately hitting the rods and cones. It's a more interesting process where the light travels through the retina and really is picked up at so-called the outer edge, the more peripheral aspect of the retina, rather than the internal part of the retina. That I found, that anatomically, that's kind of unique. So the true receptor cells are the rods and cones. We'll get to it. Rods because of their shape, cones because of their shape. But the distinction is we have about 20 or more time rods than cones, because even though our color vision is excellent, it relies on a lot of light. The rods we utilize so much because we don't require. They pick up little bits of movement, little hints of shadow and shades and silhouettes, and they're much more abundant peripherally, where cones are very minimally part of that. So, and then after the rods and cones, they intersect, they synapse with the bipolar cells, with the two ends. It acts on a dendrite with a nucleus in the middle, and those then synapse with the actual ganglionic cells. That's what becomes the optic nerve. So other than the sensor in the retina per se, there's no more sensory apparatus as those nerves make their way to the optic disc and back toward the optic chiasm, etc. So it has to pass through two layers of nerves before it reaches the rods and cones. The nerves are so fine that it doesn't obstruct the incoming rays. So they're very delicate, slender structures. They're showing it to you here. The rods and cones that you can see depicted in this black and white illustration are embedded in the posterior wall, and the vascular supply is close at hand. And so that's really where we begin to see the structural changes that are part of it, that are associated with it. And really, it's all about taking this combination of photopigments and altering their shape. That's really all it does. But what's interesting, the most interesting thing, and we talked about this in physiology, is this is a system that's designed, when it's not activated, it's technically speaking constantly polarized. So when effectively we're shutting off the circuit, and that shutting off the circuit, okay, by creating a hyperpolarized environment, actually is what triggers reception. I love things that are sort of counterintuitive. We see because the signal's turned off as opposed to the signal being turned on. That's unique areas, I think, are always things they focus on that are there. And so here, the whole idea, who wants me and why? Hold on. Okay. Sorry. And so then the other things, optic disc, blind spot, macula, fovea. I suppose you could do this, not going to do this. So the rods and cones, shape-wise, basically the variation in their proteins are opsins. Rhodopsin would be the one that we associate with the rods, where it gets its name. So the opsins individually, without getting into, I don't think you need to know the organic or biochemical basis, are retinols, and the retinols derive from vitamin A. So certainly that's the big thing. Vitamin A is interesting. Vitamin A is certainly paramount in vision. See how, vitamin A, that's, to me, is it's one thing it does most. And it's all about this idea of cis and trans bonds. Rods and cones at rest, the retinal portion, which is the part that changes, has trans double bonds and one cis. The retinal and its associated opsin keep the channel open. It's a sodium channel. Cell remains depolarized. Upon absorbing the light, the retinal goes all trans. So effectively, the way they describe it is the changeover between cis to trans. So trans for straight, cis for bent, if you will. So and certainly that's a point of emphasis. So they synapse when that occurs, when they're no longer hyperpolarized, closing the channel. The depolarization in the dark, both types of photoreceptors release the neurotransmitter glutamate on the bipolar cells. So that's the neurotransmitter that's associated, inhibiting them. So even though glutamate is usually excitatory, this is one of those oddball things. Let's say it's all counterintuitive, for glutamate, in this case, to be inhibitory other than its usual role. And so what happens is that hyperpolarizers, the photoreceptors, stop the glutamate. Okay, the glutamate no longer present, they depolarize. Okay, so it disinhibits, if you want to think of it that way. Depolarization then of the ganglion cell, action potential, etc. And again, the distinction between rods and cones, more sensitive to light, motion, particularly periphery, cones, abundant light, color, more concentrated in the fovea. Again, it's like a color wheel, blue, green, and red, and then the confluence of those and the proportions are what dictate what colors we see. When I looked at this, the physical difference, it really was a combination of the different pigments that made up the individual receptors really accounted for this. So there was a little different construction, which you wouldn't have to know, that account for that. And the brain perceives it, okay, that are there. They'll probably have a little nugget on color blindness, being sex-linked, and being an absence or a relative absence of certain kinds of receptors. I know my dad had that. I think I told you guys. Didn't keep him out of World War II. Thankfully, he was stateside. So other terms that are important. Emetropia, I beg your pardon, normal vision. Too much or too little curvature of the cornea or the lens where it's refracted, where the light is bent. Are really so when you hear myopia, like I have, nearsightedness. So I think it would be worthwhile remembering myopia correct by concave, which causes diverging by a concave or diverging lens. The light rays diverge slightly to reach the cornea. Okay, hyperopia, farsightedness, okay, corrected by convex or converging lens. Okay, and then you see presbyopia and presbyodia, and there's another question it's like why hearing is a problem. What we begin to see, the ability to accommodate, has to do with lens flexibility. The same thing associated with, we see all of these changes, and we didn't do the senescence unit. I probably should throw that in, the aging unit. Basically the little things that go wrong. It's like in the cochlea, you lose a lot of the high-pitched certain sounds you can't hear as you get older. You guys, when you have significant others, particularly women, they describe us as having selective hearing. We only hear what we want. I got smacked yesterday for that. She got a new case. No, she got a new case. We're sitting there having lunch. I went and got a sandwich from the Colonel. Okay, and she's opening up a new case for her. She's got a new phone, got a new case for it. She said, take this apart. Piece of greasy finger got it. Oh. What are you doing in my case? Case. Women. Okay, so how do we process all this stuff? Okay, I do forget what the statistic, they have a slightly different number in here. A lot of authors will tell you about half of our circuitry is about vision when it starts coming to sensory perception. Complex, argues a lot. I mean, you do it all the time without even thinking about it. I mean, so mentally, how do I know if, obviously you can tell who's here and who's not here. I know if you're in my class, all of those types of things, there's just so much familiarity and it triggers a lot of things. When we see somebody who walks in and you can tell if they look unhappy or happy, the way their gait is, all of those little things trigger stuff like that. So lines, edges, angles of movement, this information. So basically what the occipital lobe is really begins to look, it's almost I call it visual processing. So you're starting to look at lines, edges, angles, and movement. And then it assimilates this, more complex, and it looks for particular features. So then it will trigger and send that information to an area where memories are stored. So you'll have facial recognition. It's like we have our own facial recognition software that occurs. And we all have that. And all of that's part of it. So what they talk about this, they talk about, I skipped over it in neurology, this idea of parallel processing. So they're looking are processed simultaneously, looking at the form, is it moving, colors, depth, etc. Okay, it's employed by other stimuli as well. And so it was interesting that the lobe constructs somewhat of a holistic image, integrating this stored information, and the brain, like I was saying, is processing while accessing the stored information. So do you recognize it? Whose face is it? So 30% of the cortex to processing, again, it depends on the author. I don't know how current this information was from a book that's probably 7 to 10 years old. 8% processing touch, 3%. That was interesting for auditory. So maybe it's just a lot simpler to hear and understand something as opposed to whose voice it is, etc. And then they go through this stuff. So the thresholds are interesting. Okay, different kinds of threshold, minimum stimulus intensity, typically around, so required to activate a receptor about half the time, also known as the absolute threshold. In other words, 50% recognition points define the absolute threshold. And it varies a little bit, okay? So this is with the higher pitch sounds. And again, this is typically argues to less flexibility of connective tissue structures, or maybe in hearing a little bit more of a problem, not just less flexibility or vibration, a little bit more rigidity in certain structures would be another way to look at that as well. And then the difference thresholds, okay, what they call just noticeable. I thought this was interesting. So Weber's law dictates two stimuli must differ by a constant proportion. So interestingly, humans, two objects have to differ in weight by about 2%. So if I'm lifting something that's 1 pound or lifting something that's 2 pounds, I immediately know the difference. But I mean, it makes perfect sense. That's 100 pounds versus 101 pounds. You wouldn't necessarily know that it was distinctly heavier. So we've gotten pretty good with all the boxes we ship for UPS. We can lift it up and go, it's a 25 pounder. You experience is a great teacher in that regard. Types of things. I'm just saying, once a hair cell, okay, loud sounds mechanically harm hair cells don't grow. This is the nugget. The hair cells detect higher frequency sounds or the smallest, most easily damaged. So it has to do with certain kinds of sounds as well. So I didn't see this particular article. Okay. High-pitched sound is common, so it has to do with hair cells. The other thing, and you remember this from auditory, is prolonged exposure to high decibel levels. It's particularly damaging. So it's that deafness nugget that you hear so frequently. In the old days, we didn't see rock people in the studio wearing headphones until certainly the mid-70s or so. Right in front of these giant amplifiers, and they didn't wear them on stage. Then they started wearing them on stage, that type of stuff. We went to some pretty loud-ass concerts. Okay. Questions? Plenty of time. Oh, good. So these were earlier in this unit. This was the cell part. You shouldn't have any problem with these. What are composed of microtubules? Which one of these is not like the others? Which one of these is? Microtubules are eukaryotic, not prokaryotic in nature. So the flagella of a bacteria is made of flagellum. The flagella of a sperm cell is made of microtubules. always keep in mind the distinction between eukaryote and prokaryote. It's fine true with nucleolus. We know all about that. Okay. It's not bound. It disappears during prophase. We knew that. It's not it's not translation because that's a ribosome. Okay, as opposed to could it be a part of the transcription? Yeah, that would be different. And it's not existent in bacteria. You all getting these? Okay. Specialized cells, glucose transported against the gradient. That's all you need to know. Doesn't matter whether it's integral protein, whether it's a sodium ion electrical chemical gradient. Okay. If no ATP is used, by definition, okay, what's it most likely to be? That's the interesting part. Some special glucose transported against its gradient by integral protein using the energy. So effectively, to me, it is it's still active transport. It would be secondary active transport. That's one of the things you see. When you're utilizing that energy from sodium ions, a lot of times when sodium comes in, it'll co-transport. And so I'm good. Let me look at that to make sure. I don't have the answers here. I will correct that if I'm wrong. It's possible that could be, if it's using the sodium ion, then I have to correct myself and think that that's facilitated, that's secondary. Okay. But usually it's secondary active transport. I will look that up. Because I looked at some of these and I had forgotten to bring the text with me to make sure what their analysis was. I used this question on my exams. Heads up. Smooth endoplasmic reticulum is about several things. What it's about, okay, is not protein production. That's rough endoplasmic reticulum. So, which one is expected to contain the most smooth endoplasmic reticulum? So, it has to be a cell. And a zygote is certainly a cell, but I don't know that there's any distinction between that and smooth endoplasmic reticulum. A sperm is typically, I would exclude that because it's really not a true cell. In that sense, it's limited, it's mostly genetic material. Certainly would not have a lot of smooth endoplasmic reticulum. And remember, from the mom is where we get the internal structures, the cytoplasmic structure. Islet cells, definitely not, because it's a protein factor. So, liver cell. Liver cells are something that are very distinctive. They do a lot of detoxification. That's fat processing for the liver. Tight junctions. To me, tight junctions, what do? Yeah, they're the principal ways that we connect adjacent cells. There's only two ways we do that. They form a barrier. To me, fluids. Tight means watertight. They have the greatest strength of all cellular adhesions. Is that true? Which one does? Desmosomes. So it's only 1 and 2. Thank you. So tight junctions are strong, not as strong as desmosomes. Gap junctions really aren't strong at all. They're protein passageways. A little opening on like a perforation on one side married to another side. Not membrane bound, easy. Okay. By definition, all of these are membrane except for a ribosome. So the mistake students would make, well, there's bound ribosome. It doesn't say bound. If it said bound ribosome, we wouldn't have an answer. But they're not going to give it away and say free ribosome. Here's the liver detoxification, and what's the organelle that does that? Again, smooth endoplasmic. Primary lysosome with a few. What happens in a lysosome? Acid hydrolysis. So pH will drop and actively pumping protons into the interior. Not in the exterior, it's what's happening in the lysosome. Okay. The I questions, the special sense questions. So you should know the normal, which of the following gives the normal direction of a signal, dendrite cell body to axon. Typically would be normal. Okay. Axon to cell body to the dendrites. Usually not the typical transmission. When the normal direction, you have to think that way. There. Novocaine. As we actually mentioned how Novocaine worked here in class. Physiology. How does it work? And if you didn't know, you could probably figure it out. So, you're just okay. Does it, if it stimulated voltage-gated channels, would that make the signal more likely or less likely to be? Because basically you're blocking the signal. Think of that. Voltage-gated channels in a nerve make it more likely to fire. Yeah. Chloride ion efflux during an action potential. Again, it would, if anything, it would make the interior more positive, more likely to fire. Schwann cells wrapped, I don't know, they wouldn't have to do anything. Yeah. Sodium voltage gated channels, oh yeah. And that's the mechanism. We used to think years ago, I would teach that we know better now, that it displaced the calcium. That's what the original thought, that it was a displacement reaction, but now we know it's more about the voltage gated sodium channels. Cell membrane, slightly passive, permeable to potassium ion. If the membrane were to become suddenly impermeable, the potassium couldn't get out, but it still had a sodium-potassium pump, what's happening on the inside? Is it becoming more or less positive? If there's more potassium on the inside, because the pumps are pumping it in, and the potassium can't get out, it's got to be more positive. And if that's the case, the ion concentration would have to increase. Acetylcholinesterase inhibitor and a cholinergic synapse. What would happen, okay, to the activity of the postsynaptic neuron? So if there's more of it, which likely it would degrade more rapidly. It wouldn't decrease because it would bind, no. It would increase because acetylcholine would be produced more rapidly, no. It would increase because it would decrease because acetylcholine would be degraded more rapidly. Oh, it's acetylcholinesterase inhibitor, sorry. Read the question, Lou. Okay. So, and that's basically how acetylcholinesterase inhibitors, that's another test question. How do they work? You know, like nerve gas type agents or other agents that increase it. So it would increase it, okay, because acetylcholine would be not produced. Acetylcholine would be degraded more slowly than oh, read the question. White matter is white because it's not white. Myelinated axon. Jumping, saltatory conduction, ratio of sodium to potassium ions transferred, pump out of and into the cell. Okay, so you pump 3 sodiums out, 2 potassiums in, consistent relationship, and which following is found in vertebrates but not in vertebrates. Ta-da! Good old dorsal hollow nerve cord. Basic, biology, high school biology. Okay, we're good. We're done. You're done with me for a week. Actually, 2 weeks. Just saying. Goodbye out there if you're going to listen virtually.